Okay, so I mentioned before, a mutation is a change in the sequence of nucleotides. And it could be a change within the DNA of the cell, or it could be a mistake in transcription. Now, if it's a change in the DNA, then it's going to be made in each and every time that RNA is transcribed and translated. That's going to be obviously more serious than a one-time mistake in transcription, which is only going to affect proteins made from that one RNA. Now, mutations can also happen in somatic cells, for example, that might cause cancer. Remember, most mutations don't cause any problems at all. But if you have a mutation in the cell cycle control center, in the genes that control the cell cycle, then that will could cause unrestricted mitosis, which is cancer. If the mutation happens within the cells that make the germ cells, the sperm and eggs, then that mutation will be passed to the next generation. Okay? Now, some mutations are advantageous. They increase production of ATP or something like that. They create a new capability. The ability to digest lactose as an adult is what we call a gain of function mutation. It's an advantageous mutation. People who do not have that mutation can't break down lactose as adults and can't drink milk and therefore can't get nutrition from milk as adults. Some mutations are disadvantageous. They decrease a capability or productivity. Most of them are neutral because they don't actually cause any change in the protein, or they're in a non-coding section of the DNA that doesn't get expressed anyway, so you could change it and it doesn't matter. It's in an intron. It's going to it doesn't get translated. So who cares? All right, so let's talk about types of mutations because there are different types depending on what happens when the mutation happens. So the easiest or the simplest is just a substitution where, for example, this one's supposed to be a G. Instead of a G, an A was inserted when the DNA was copied. Now we have a different amino acid coded for, and every time this gets translated, it's going to put the wrong amino acid in there. And again, this might be a huge problem or it might not. There are some proteins that can act perfectly normal with several substitutions within them. Sometimes one substitution can make a huge difference. Sometimes it doesn't make any difference at all. All right. A deletion is literally where one amino acid gets deleted. And the problem with this is that, remember, codons are 3 letters, and there's no placeholder to say there used to be an amino acid here. So every letter of all the words after this mutation shifts to the left. So instead of the codon being UUU, the codon is now UUG. And the next one, instead of being GGC, is now GCG. So it's going to affect all the amino acids from that point on, which could be a huge change in the protein. We could have an insertion, which is going to also change all of the amino acids in the strand. Same thing, instead of just doing UUU, it inserted a G in there by mistake. Anything that causes a mutation in DNA is called a mutagen. Gen means creation. Okay, Genesis, the book of creation. Okay, so mutagen means it creates a mutation. Now, there are physical sources of mutations, such as UV light. You've heard that sunlight can cause skin cancer because it causes a mutation inside the DNA, inside skin cells. X-rays can also cause mutations, which is why they put a lead apron on you whenever you get an x-ray. That's to limit the amount of your tissues that are exposed to those x-rays. There are also chemicals such as benzene that can cause mutations. If a mutation or if a mutagen causes cancer, then it is called a carcinogen. And all of these can also be carcinogens in addition to just being mutagens. Not all mutagens are carcinogens. Remember, most mutations aren't good or bad, they're just nothing. Okay, so that's the end of that. I wanted to go back. You know what? Hang on. I'm going to go back and do one more thing. I got to go all the way back here. Wait, where'd it go? Here we go. I want to talk about one more thing here. Okay. You remember I said that thing about how we could make multiple proteins from one gene? It has to do with this. Imagine if instead of this always being an intron, what if sometimes this was an exon? Then we would have a different mRNA strand, and it would make a different protein. And that actually happens with a number of our genes. The nucleus can actually splice out different sections depending on what protein needs to get made. And that is why our genome is smaller than expected, because in many cases, one gene actually provides the instructions for multiple proteins, depending on how it's spliced. How cool is that? Okay, now I'm done with this chapter.